and that we can get going and get started. So it looks like the sound is coming through clearly, the picture's coming through clearly, so I think we can get going. So I'm going to reduce that screen, and we've got left this big screen with the first green slide. So we're going to be doing a series of 4 lectures on the reliability of genetic tests for personalized nutrition. Like I said, I'm Duncan Carmichael, and we are going to be doing it over a period of 2 or 3 months, and each lecture should be 1 hour long. Hopefully it's going to be interesting. It's been fascinating putting the lectures together. It's stuff that we've been talking about and talking on for the last couple of years, and it's a topic that is highly, highly topical. As you might hear, I'm a little bit bunged up, a little bit block-nosed. That's because we're right in the heart of winter in Cape Town. Megan and I will be flying back to beautiful London in about 3 days' time, so if anyone's going to be at the CAM conference in London on Saturday, we'll be flying in for that. We're expecting sunshine, so if you can please organize that for us, that would be great. So we're looking forward to coming back there. What we've been doing out here in Cape Town is setting up my DNA Health, which we'll be doing for the next year or so, and bouncing back between London and Southern Africa. So just a little bit about me very quickly, just so you understand where I'm coming from. I'm essentially a clinician. I'm a clinician who trained as a GP in the UK. And you'll notice from my accent it's been somewhat watered down. I left the UK initially when I was 6 and came out to Southern Africa, and I've been going backwards and forwards ever since. Since then I went into aesthetic medicine and then to what we call anti-aging or healthy aging, which really started off as hormonal medicine and evolved into nutritional medicine and then into genetic medicine. So genetic medicine has become a focus of huge interest for me and for many others over the past few years, and particularly in this 2 years, the relevance of it to the sugar and fat debate, which we'll get into in the next couple of lectures, I think is fascinating. I think that genetics and nutrition hold many keys to that topic and that debate, and hopefully we can enlighten on that in the next couple of lectures. So I work with My DNA Health. I'm founder and chief scientific officer for it, and we've had a lot of fun developing what we hope is a very balanced genetics testing program for people. We focused at the nutritional level, and nutritionists who I feel with functional medicine are in fact further ahead than the doctors I lecture to. I'm not just being polite, I really feel that. I think functional medicine and nutrition has really made massive strides and is leading the way in many things. So our focus really is with nutritionists, and we hope to get feedback from you about how relevant you are finding our tests. We're hoping that these are going to be interactive. I won't be speaking too much about My DNA Health at all. This is a scientific talk, but it is important that you understand and know disclosures. So we have 60 slides to get through, so we're going to get cracking. But before we start, if we can have a quick poll. So Amy, if you could initiate that poll, and it will just be interesting for us to know, are you using genetic testing in your practice, or are you not using genetic testing in your practice? So we can wait for those answers to come through, and then I'm going to reduce that from my slide, and we'll go back to that. All right, so Megan's saying that 20% say yes, you are using genetic testing in your practice, and so far 80% are saying no, you're not currently using it, which is interesting and important. So the way I'm going to pitch the lecture will be more to the fact that we're not using genetic testing that much. There will be, it very much is an introductory lecture, but it does speed up as we go along. So this is today's lecture, with the overall topic being the reliability of genetics testing for personalized nutrition. So it's the introduction to genetics. Webinar 2 will be genetics and fat metabolism, and we're expecting that to be in 3 weeks' time. The final date isn't quite set. then genetics and sugar metabolism, which is even more interesting, and then we get into the wonderful, fascinating topic of the methylation cycle and the liver pathways, particularly for the steroid hormones. And that should be, hopefully, a fascinating talk. So today's agenda, starting with the future of genetics, just a couple of slides of some of the interesting things we can look forward to in genetics. The history of genetics, which evolves into the theory of DNA in genetics, and central to that will be epigenetics, and finally genetics, DNA testing, and practical application. So the message that I would like everyone to take away from today is your genes are not your destiny, but they are your tendency. So as we go along, what we'll do is, if you have questions, please type them into the box that you will see on your computer, and there is a area there for questions. Megan here will field the questions and type back answers as she can. We need to keep the whole lecture evolving, and so as we go through it, if there's time at certain spots, I will stop and answer as many of the questions as I can. But we do want to keep the entire lecture going and going through the hour and not go beyond the hour. And then if we have questions after that, it would be great to field them then as well. All right. So the future of genetics. Something that I find fascinating, the immortal jellyfish, the Turritopsis. This is a tiny little jellyfish. It only grows up to 4 centimeters long, really is small, and the only species known to live forever. And how it does it is that the mother jellyfish mitoses or buds off a polyp, and that polyp then grows through to the medusa size and with exactly the same DNA as the mother, and continues to grow from there. The mother then will make more polyps, and the mother will live forever, potentially, until it gets destroyed by something else. If it's in danger, it can shrink back into a polyp shape, and then when the danger is gone, it can, danger being toxins presumably in the water, it can evolve back into a fully grown jellyfish. So a fascinating species, and Professor Shin Kubotu in Japan has been studying this for the last few years and is quite sure that the genes of the tiny immortal jellyfish could hold the key to longer life for humans, which is a topic that's always interesting. Whether we agree or disagree with the topic, it's always an interesting topic. And on a similar vein of things, Berenice Syndrome, which is sort of a version of progeria. It's an adult problem, an autosomal recessive genetic mutation, which causes adults to age rapidly. Progeria starts in children, and is also genetic, but has a different protein that's affected. So heterochromatin is the protein that's affected in Berenice syndrome, and various studies and testing on heterochromatin and the DNA coding for it, scientists feel that there may be some breakthrough in how we understand the aging process. So I think that those are two fun topics to look at in the future. But really what we need to do is start with going back to the beginning of things. And there's no problem really. We're going all the way back to Gregor Mendel in 1850, who I feel is the father of modern genetic testing, who proposes gene theory from his studies on sweet peas. Now I'm going to bring my pointer out here, and I've also been warned I mustn't use my pointer too much or it makes people a little seasick, so I'm going to try and be quite conservative with it. So there's Gregor Mendel at 1850, and it took us 100 years to 1950 before we had the discovery of the double helical structure of DNA. And from there, genetics and gene testing accelerated quite quickly. It took only another 50 years until the entire human genome project was completed. And since then, we've got into epigenetics, and things really have accelerated. So that really is our timeline of the history of genetics. So Gregor Mendel, in the 1800s, an Austrian monk, as we all know, was a sweet pea plant breeding monk. It seemed like quite a gentle fun life that he had, and he had plenty of time to grow the sweet peas, and observed, really by observation, which was quite clever, observed the different principles. And these principles we use up to today. They haven't changed. And so his Punnett square was his most famous contribution, where if you have parent 1 and then parent 2 at the top, and parent 1 with a yellow sweet pea will contribute an allele from one parent, which is the yellow dominant allele, and an allele from the other parent, which is the green recessive allele. And parent 2, which is a green pea, will have a recessive and a recessive, making a green sweet pea. And that will combine with that to give a 25% chance of a yellow sweet pea. That will combine with that to give a 25% chance of a sweet pea, and likewise for a green sweet pea, that allele, and likewise for the fourth square. So for most of genetics, we can work out the likelihood of the future generation, and in this case, sweet peas being either yellow or green sweet peas from that Punnett square. So that was his one contribution, and his other contribution was the dominant recessive heterozygous homozygous terminology. So that is a dominant sweet pea because the yellow is the dominant allele. That is the recessive sweet pea because both of those are recessive alleles. And then we have heterozygous, where we have one of each of the different types of alleles, and then homozygous, where we have identical alleles making up the gene. So dominant, recessive, heterozygous, and homozygous was the other terms that were brought in. And then from there we went on to the concept of the genotype, which is the gene, and then the phenotype, which is what it turns into in the body. So we would expect that if our genotype is a homozygous dominant BB, then we would get a grey rat. Heterozygous, with a big B and a little b, will get a grey rat. And if only we have a homozygous recessive, little b, will we get the gold-colored rat. So that genotype, phenotype expression of things became the next term that was quite useful. And so a fun way for us all to have a look at that is with our earlobes. So if you have a feel up to your left or right earlobe, is it detached or not detached? So most of us should have a detached earlobe. And we'll be homozygous, dominant, or heterozygous for having that. And then 25% of us could have an attached earlobe. And that would be the homozygous recessive coding for that. So these alleles will sit then on chromosomes, one chromosome from the mom, one chromosome from the dad. And the allele at a certain point will be for phenotype A and perhaps for phenotype B. And that really builds up the chromosome placement of the alleles on the chromosomes. So at conception, we will have an egg and a sperm, each with 23 chromosomes. They will combine together to form 46 chromosomes, or 23 chromosomal pairs, which will be replicated through our entire body. So then we get to what I think starts to become the fun bit, which is how these chromosomes are coiled, how this information is put together. And it really is remarkably detailed. So if we have a chromosome here, it is made up of these supercoils, so coils upon coils. And these supercoils then are, if you unravel them further, you'll get the coils, which is where you have the histone protein with the DNA wrapped around it. And if you unravel that further and take the histone out, you can then start to read, transcript the DNA. And that histone-released transcriptable DNA is then called the DNA double helix, that thing that was discovered in the 1953. And that double helix, we then discovered, was made of base pairs. And how those base pairs are formed is you'll have a phosphate group on the edge, and then a sugar deoxyribose group just inside. There's a deoxyribose, and inside of that, one of the four very famous purines or pyrimidines, so adenine, guanine, cytosine, or thymine. And those, adenine, guanine, cytosine, and thymine, are the base pairs, which match all the way along the double helix of the DNA. So that's DNA unraveling, and that's then new DNA forming over there. And that's an amazingly tightly coiled system of information. It's absolutely enormous, and how they managed to read all that to get genes and gene testing is unbelievable. So how it's read, these purines and pyrimidines, these base pairs, it's read in the form of a codon. So a codon is really a 3-letter word. So mat, cat, hat, at, well not at, but the rest of them are fine. And so we have these nucleotide bases, the letters adenine, guanine, cytosine, and thymine, and they form these 3-letter words called codons. And each codon will code for an amino acid, so that one is coding for proline. We have 21 amino acids, and these amino acids combine to make the proteins, and the proteins perform all the functions in our body. So there we have the codons, the words that make up the language, which is the DNA of who we are. So we can get the concept that codons make up the musical script of who we are. And that script is kept safely in a book of coiled chromosomes, but it still needs to be read. It still needs to be read and played and come to life. So with that in mind, sometimes there's a spelling mistake. And that spelling mistake is called a SNP, a single nucleotide polymorphism. So a single change of one nucleotide. And if we see that the sentence, the cat sat in the mat, would make sense, but if we misspelled cat by one letter, we would get the tat sat in the mat, which makes no sense. So similarly, a SNP would be a change of one nucleotide to get a change from this message to this message to this message in different people. So that single nucleotide polymorphism is the spelling mistake that makes variety. Now we'll see that sometimes a SNP can make absolutely no difference at all. So for glycine, you could have the codon GGU, but you could quite comfortably change the U to an A or a C or a G, and it will still code for glycine. The codon for lysine is AAA, AAA, but if you change that last A to a U or a C, it will change it to a different amino acid, asparagine. So some SNPs will make zero change and some SNPs will make significant change. So the SNP is very important, leading into improvements in our genetics or to disease. And so one of the most famous SNPs has got to be the sickle cell anemia disease SNP, where in this series of codons, there's a change of one nucleotide changing from A to T. And that change from A to T makes all the difference and changes the red cell, the red corpuscle, from that shape to that shape, which is obviously a survivable change up in Central Africa when you want to improve your chances of survival. So that SNP change for the sickle cell anemia can be written in different ways. So we could write it so the A has changed to the T. Well, it wasn't very good writing, was it? There we go, there's a T. So the SNP can be written as A is greater than T, where A is the original nucleotide and T is the wild type or the abnormal one. It can also be written as the amino acid that it's coded for. So at position 27, glutamine is the original one and valine is the wild type. It can also be written as HBB, which is the gene test for sickle cell anemia, A, which is the original amino acid 677, and then T, which is the changed nucleotide. So there are different ways of writing it. Generally, when people are talking about a SNP, if we take the methyl tetrahydrofolate, the MTHFR gene, then it will be written as either heterozygous or homozygous. So we may be healthy, having that CC allele. We may be heterozygous unhealthy, having the TC, or homozygous unhealthy, having the TT. But what is written is, if you're heterozygous, if someone says you're methyl tetrahydrofolate heterozygous, it means you're heterozygous unhealthy. they say you're methyl tetrahydrofolate homozygous, then what they mean is that you are TT unhealthy. So if they say you're homozygous, you're unhealthy. Heterozygous, you're heterozygous, unhealthy. And healthy or normal is just assumed as normal. So that's just a convention of when people are speaking about it. That's how it's expressed. So with all of that information needing to be collected, scientists around the world collaborated to start the Human Genome Project. Started in 1990 and unbelievably completed in April 2003. So it was an amazing collaboration, identifying 20 to 25,000 genes and the sequences of 3 billion base pairs in the human DNA. And really at that stage we thought, well, we got it. We've got the book, it's all written down, and we know which genes are what, and everything should be easy now. Except that the problem was we then discovered that we had the epigenome. And the epigenome was a layer of information on top of the chromosome that affected the chromosome and the information that came out of it either positively or negatively. So the Human Epigenome Project was then started to discover, gosh, what is all this information on top of the information that we already know? So really, if we look at it, we will have everything that we do, everything that if we breathe in, we worry about, we ingest, everything affects the surface of the genome. And that surface of the genome is the epigenome. Epigenome really means the outside, the all around of the genome. So information will affect all around the outside of the genome, and that information that affects it will either slow down or accelerate the expression of the information inside the genome. So that's the epigenome. So more specifically, what we'll find is that methyl groups become so important, and methyl groups attaching to the unraveled chromosome will encourage reading of the codons in that particular area. Alternatively, methyl groups or toxins attaching to the histones could slow down the reading, could slow down the unraveling of the histone and slow down the reading of the DNA. So that is the epigenome. So really, two things from that. Methyl groups and methylation become extremely important for the reading of DNA. And without healthy methylation, the reading of DNA really becomes quite a stunted affair. And that secondly, that this information encoded around DNA, around the genome project of accelerating or slowing down information becomes absolutely vital. So if we were to understand it with a different analogy, if the DNA of the genome is your musical script, then the epigenome is how the music is lifted off the page and played. It's the orchestra. Is it played softly? Is it played loudly, slowly? That's the epigenome, and it's so, so important. So this Human Epigenome Project is ongoing, and a lot of the discussion about the relevance of DNA testing is around the epigenome. So, for example, DNA methylation, as we know, is central to the epigenome. Our ability to methylate is central to the epigenome, and what we worry about is central to it. And one of the best ways that have been shown of how the epigenome affects our DNA is to look at identical twins. And with these photographs and many others in the series, a Chinese photographer showed the difference after the age of 50 between different identical twins in China. There have been other studies along the same sort of lines, but it really shows that if they have identical DNA, then the difference that they evolve through life is down to the epigenetics, what's influencing the outside of the gene. And you can see that the changes can become quite pronounced through life. So to learn more about epigenetics, I'd really encourage you to go to this YouTube clip and watch it. And what they'll show you in it is about 13 minutes long, so it's not too long. So I'll stay on this slide for a moment just so you can write this down, and I'm sure that this will be forwarded to you by Bernie afterwards. So what you'll find here is that the scientists will show that the agouti mice, genetically identical agouti mice which contain the agouti obesity gene. And so if you take a mother who's either obese or not obese, doesn't really matter, and feed that pregnant mother a toxin, then all of the offspring will become obese, have heart attacks and cancer. So the explanation is that toxin affects the agouti gene from the outside, from the epigenetics, and all of the offspring are obese. On the other hand, if you feed even an obese mother plenty of methyl supplements while she's pregnant, so her greens, her kales, then all of the offspring will be thin and healthy. So that agouti gene is overridden by the methylation. So that was really seen as a seminal study supporting the importance of epigenetics. That's a very good YouTube clip that explains epigenetics. A term came from there called the exposome, so the totality of how the environment interacts with our gene expression from conception to grandchildren's genetic expression. And we'll get into grandchildren's expression in a second. So if your genes are the loaded gun, your lifestyle pulls the trigger. There's another analogy to look at. So the FTO gene is a good gene to look at, which really expresses some of the power of epigenetics. So the FTO gene means that you're predisposed for the rest of your life to be obese. However, by walking every day, you reduce that risk factor by 30%. Anything in yellow at the bottom is the study, so that's the study at the bottom that was taken to show this. And then by eating your greens, so the methyl supporters, you reduce the risk further. So that really is important when it comes to how we explain to people their gene tests. So obviously it's absolutely nonsense to say you have this gene and therefore you're going to be obese. It really just isn't true at all. But we would explain it to them that you have this FTO gene, there's a risk of being obese, but that it will either be switched on or switched off. And being a low penetrance gene, and we'll get into that later, the chances are it's switched off. And there's various things that people can do to make sure it stays switched off. So if you have that gene, it is healthy for you to walk every day, as been shown in the studies. It's healthy for you to make sure you're getting your methyl supporters, your green vegetables. So giving the positivity of the gene result is much more important than the negativity, because the chances of the negativity actually happening are really quite low. So further on the topic of epigenetics, the Avon longitudinal study in the UK, so that one of the things that showed, and it's an ongoing study with many things it shows, but one of the things that shows is that parents who smoke, the children have a higher tendency to obesity. So what your parents and grandparents did affects your genes today. And Pam Peake, who's quite hilarious, I think she's a New Yorker, takes this topic and expands on it beautifully, again in another 10-minute YouTube clip, and it's well worth watching. You'll have a giggle, and she explains this tendency of your parents, of your grandparents. There's studies from the Danish epigenetic studies which explains that further as well. That's well worth a watch. So epigenetic effect on coffee, the CYP1A2 gene expression. So you'll find that there's been a tendency with gene companies, commercial gene testing companies lately to say that if you CYP1A2 homozygous, that means you've got the wild type, you've got the bad gene, you're CYP1A2 homozygous or even heterozygous, then you shouldn't drink coffee. So let's look at that. So in this study, the objective was to investigate the influence of coffee consumption on CYP1A2 enzyme activity. So CYP1A2 is the gene, and that codes for the CYP1A2 enzyme, which is the active protein that does the work in the body. Generally, the gene is written actually in capitals, and the enzyme is written in small cursor and small case. So 149 Swedish volunteers using a detailed questionnaire, oral concept of use, habitual coffee consumption. So daily coffee consumption of at least 3 cups a day significantly increased CYP1A2 enzyme activity in all the participants. So their conclusion of this is that habitual heavy coffee consumption increases CYP1A2 activity, i.e. the coffee will have an epigenetic influence and upregulate or turn on further the CYP1A2 enzyme. So the interpretation of that is that unhealthy carriers of CYP1A2 gene should drink coffee, as it activates the gene and helps them to remove coffee and other toxins more effectively. Now we get to this study, and this study again looked at the CYP1A2, so the cytochrome P450, the CYP1A2 gene and the enzyme. And the concern was that people who drink coffee who are CYP1A2 positive and have hypertension could worsen their hypertension. So the data showed that the risk of hypertension associated with coffee intake varies according to CYP1A2 genotype, and that people who are CYP1A2 positive or homozygous, that they should not drink coffee as it leaves them at risk of hypertension. So interesting. 2 studies, one of them saying if you're CYP1A2 homozygous, drink coffee because it will upregulate the CYP1A2 enzyme. The other one saying CYP1A2 homozygous positive, don't drink coffee as it raises your risk of hypertension. So 2 completely opposite interpretations of that gene SNP that could be quoted. So within epigenetics we get Nutrigenomics, and maybe I'll just pause here at this stage. Megan, are there any questions I should go through just to get people to get their breath? Okay, here's a question. All right, so question for Doctor. Can you therefore assume that a SNP for Serbs and Swedes are similar all over the world, i.e. one size fits all or does not? I'm not quite sure what that's meaning. Could you, whoever put that question is, could you put that through again? Just put it in different words. I'm not entirely sure. I assume that the SNP for Serbs and Swedes are similar all over the world. No, I wouldn't assume that the SNP for Serbs and Swedes are similar all over the world. It's going to be, so that really goes back to this epigenetic effect of coffee over here. No, it won't be. The SNP's going to be different. Right. So how should we behave about coffee then? So my opinion is that if you are CYP1A2 homozygous and you have hypertension, then maybe there's a consideration not to drink coffee because of a further worsening of your hypertension. But hypertension can be measured and monitored and controlled, and really none of these low penetrance genes should be scary things. And I think to tell someone to stop something on the basis of a small study like that for something that's not particularly dangerous, I think is a little irresponsible. I very much like, and we'll see later with some other tests, I very much like the approach that we can upregulate and downregulate some of these genes, and particularly the cytochrome P450 genes. And so myself, I'm CYP1A2 positive. I started off by drinking 1 coffee and then went up to 2 coffees. There's been absolutely no problems with my blood pressure, and my ability to deal with coffee has improved a lot. So I have improved my ability with that enzyme. So I'm hesitating because I'm reading another question. What if you're heterogeneous, I think heterozygous, should you drink coffee and do the same lifestyle to apply? I think the point is that I'm more in favor of this study and less in favor of this study. And I know there's been a lot of trumpeting about don't drink coffee if you're that. I disagree with it. And I disagree with it on the basis of the study. And I really think that upregulating enzymes is important. If you are hypertensive and you are homozygous for that, well then maybe it would be a good idea to back off from it. But for the rest of us, if you're homozygous for that, I think 1 to 2 coffees a day, and you will see how you improve. You drink 1 coffee, if you're wired for 4 hours afterwards, well then your enzyme isn't upregulated. You're not processing that coffee out your system. But if you drink a coffee and it feels normal, then that's great. So start with half a coffee and build up slowly from there. So unless you're hypertensive, I disagree, and I think it's a little bit of scaremongering. And there's not a lot of studies to back that up. So it's small studies, and I don't like it. I much prefer this approach over here. All right, let me move on. Nutrigenomics. So genetics plus nutrients make us what we are today. So I see nutrigenomics as being very much a subcategory of epigenetics, and it's the nutrients plus our genetics and how the nutrients influences us and who we are. So if we look at the most famous company, 23andMe, and they have done some great work. They've also been in the press. And they do comprehensive testing, but they've been in the press for the wrong reasons lately. And the wrong reasons really sort of goes into the topic that we're starting to talk about, and it's cancer scaring. And that really, with these low penetrance gene tests, we really can't go around saying that you're going to get cancer, you're going to be obese, you're going to have a heart attack. That really is just wrong. So the FDA suggested that reports a false positive for a cancer gene, that by reporting that you may get cancer from these genes, it could lead to a patient undergoing prophylactic surgery, chemoprevention, intensive screening, or other morbidity-inducing actions. While a false negative could result in a failure to recognize an actual risk that may exist. And this comes back to the point that gene testing is just gene testing. It's not phenotypically who you are. So I want us to go into the high penetrance and low penetrance genes so we can see that. So there's no guarantee that any one gene will be expressed phenotypically. And really the message again of today is that your genes are not your destiny, but they are your tendency. It's a good map for us to guide us along, but it really is not our destiny. So if we look at high and low penetrance genes, so genetic architecture of cancer risk. So at the far end over here, a high penetrance gene, very, very rare, would be BRCA1 and BRCA2. And then all the way down the other end, very, very common genes, but with very little risk, would be, for example, CYP1B1 and other cytochrome P450 gene tests. So we look at the very famous case recently of Angelina Jolie, who had mastectomies and then oophorectomies to prevent her genetic risk of breast cancer. What's that about? So that was about the high penetrance genes. That's about the BRCA1 and BRCA2 genes. So they follow the Mendelian pattern. And there's first generation, second generation, third generation. Across through the line through there means that person passed away. And so you'll see that in every generation there's someone who actively has the problem. So the prevalence of BRCA1 is about 2.5% of the population, and BRCA2 gene is about 2.5% of the population. So it's a very small percentage of the population who will have this gene, and the gene testing is not cheap. But if it's in your family, it's certainly worth testing for. It's important to test for. And then what do you do about it? Well, that answer comes from this section over here. It's a high penetrance gene. That means the chances of the gene actually causing breast cancer are very high, 56 to 87% if you have BRCA1, and 56 to 87% chance if you have BRCA2. BRCA2, by the way, is also positive for prostate cancer. There we go. so a risk factor for prostate cancer. And also what's interesting is, why is there such a variation between this penetrance? 56 to 87% is a wide variation, and it's believed to be the epigenome. So even with high penetrance genes, if you're on the pill, if you're smoking, if you're stressed, that makes a big difference. So what causes the variance of gene expression? So nutrigenomics, the effect of the nutrients on our genes, the foods that we eat positively or perhaps negatively. The epigenetics, so the influence of the entire world on our phenotype, the positive and negative messages we get all the time. And then other genes. So for example, with our sugar and fat metabolism, we may have a healthy PPAR gene, but 3 other genes, ADBR2, TCF7L2, and tumor necrosis factor alpha, could all be homozygous, and they could be conspiring to give us sugar diabetes. So one gene on its own, PPARG, if it's healthy, does not mean that we won't get sugar diabetes. So to give a bit of perspective on low penetrance genes, I think this is quite useful. So this is a slide we'll get into in more detail in the 4th seminar. And really, if we can just look, that's estradiol, E2, that's estrogen coming into the liver. And we know when it goes through the liver, it needs to go through phase 1 and phase 2 detoxification. And it does it via 3 pathways. And here's our cytochrome P450 pathways, and here essentially is our COMT, methylation, and glutathione pathways in the phase 2. And then it gets passed out through the body. So in the phase 1 pathway, if it were to go down this pathway, that's first prize. It's very low risk, there's really no problems, and that's really where we want the estrogen to go through and out. It's safe over here, it's safe over there, it gets stuck on hurdles in the middle. If it goes down the CYP3A4 pathway, well, if it gets stuck there, that's an unstable estrogen molecule, and that can lead to breast cancer. I think often more importantly, if it gets stuck down this pathway over here, the 4-hydroxyestrogen, that's an unstable molecule, and that can lead to breast cancer. So this enzyme and this enzyme is central to whether the estrogen gets sent out of the body or doesn't get sent out of the body healthily, or whether it gets stuck in that position or that position. So if we look at Premarin, one of the HRTs, it was discovered after the Women's Health Initiative that Premarin in fact comes into the estrogen detoxification pathway halfway down this unstable pathway. And so people who have, or women who have problems in this pathway, will have an increased risk of breast cancer taking the Premarin. So it doesn't even have a chance of getting down this pathway, it comes into the pathway over here. On the epigenetics of it, there's various nutrients that will influence positively or negatively. So red is an inhibitor, green is a stimulator. So over here on CYP1B1, so part of the pathway that we want to inhibit. We don't want estrogen going down here, we don't want it going down there, we want as much as possible going out through there. So this CYP1B1 is inhibited by these factors, so polyphenols, resveratrols, flavonoids, green tea. And this pathway is activated or turned on or switched on by cigarettes and plastic toxins, dioxins and PCBs. So we can influence hugely epigenetically with the nutrients or the toxins that come into us this pathway. And we can influence hugely this SNP and this gene and this enzyme. So if you are CYP1B1 homozygous, this probably, and it's very difficult to get figures on this, but this probably increases your risk of breast cancer by perhaps 2%. So it's not 50 to 87%, it's a very, very small increase. And this is modified better or worse by your environmental exposure. So the point is that if someone is CYP1B1 positive, homozygous positive, they are not going to get breast cancer, they're low risk. but they have a very small increased risk of breast cancer, and there are nutrients they can take to decrease their risk and toxins they can avoid to decrease their risk. And that is a much, much healthier way of explaining things, an accurate way of explaining things to patients, rather than saying you're CYP1B1 positive, you need to be aware of breast cancer. So I hope that makes sense, and I will go into it again in the fourth seminar. Megan's just writing something down. Good question. What would you advise your wife if she has the BRCA gene? That's a very difficult question. And I think that's a discussion that one needs to have before doing the blood test for the BRCA gene, is if it's positive, what would you do? And there really generally ought to be counselling facilities available before tests like that are done. But if the BRCA gene were to be positive, I would then look at her family and the expression of the gene in that family. So if there really wasn't much expression of the gene in the family, then although it's a high penetrance gene, it's going to be a low risk gene, and that may influence my decision as to whether one would do a mastectomy or not. If there's high penetrance of the gene in the family, so that every generation there's a direct line of women who have breast cancer, and it's her mom and her grandmom and her aunts, then I think is much more of an argument to say it really would be a good idea to look at mastectomy. But again, that's a question that should be posed before the testing happens. Okay. So to repeat this slide again, Nutrigenomics, your genetics plus your nutrients makes you. And I think we're coming to the end, actually, which is quite good timing. We've done 50 minutes. So genes in our environment. Our genes determine who amongst us is susceptible to the particular condition. And our environment, or the epigenetics, determines who amongst the susceptible will develop the disease. So our environment is largely determined by our lifestyle choices, and nutrition is a major component of our lifestyle choices. So there's various studies showing that various nutrients will make various benefits to various problems in the body. So here's one showing that eating fish decreases inflammation in people with high TNF-alpha expression of the TNF-alpha gene. Now I just wanted to show this slide, which is really the only one from my DNA Health, so from the company, to show the importance of taking into consideration the epigenetics. So if we look at, this would be a slide, or this would be a field that you as a nutritionist would see at the back end of your patient's results. And you'll see that over here are a list of gene tests: ADBR2, Actin3, which is the muscle fast or slow twitch, COMT, which is the liver methylation, and MTFRB, which is methyl tetrahydrofolate. And so these two being on this slide, the liver detoxification methylation pathway tests. So let's talk about liver detoxification methylation. So both of them are homozygous. So one would suggest that the person has a problem with detoxification. However, what the patient would also have done, and you'll see up here, are the questionnaires. And they would have done a liver function questionnaire. And from that, one will see that they didn't get a particularly high score as regards their questionnaire for toxins and toxin load in the body. So you've got someone who is heterozygous for COMT and MTHFR, but their questionnaire is saying that they're not particularly toxic, they're actually quite healthy. So how do we approach that? And we approach it, I approach it, by telling them that they need to be aware of this. They need to be aware of supporting this. And that through life, even if they're not feeling toxic at the moment, there's no fatigue, there's no inflammatory symptoms, there's no histamine symptoms, there's no accelerated aging, that at the moment they may be quite healthy. But every year that passes, there's more and more chance of an expression of these genes. So if you remember the slide with the identical twins, through life, because of their environmental exposure, they diverge. The one doesn't look or is as healthy as the other. So at the moment, their liver function test or their liver function questionnaire may be healthy, but they need to keep an eye on things. So an example of that would be an 18-year-old young man who comes through to you, competent MTHFR positive, but very healthy. 2 years later, after 2 years of drinking alcohol at university, you find that they come back to you, they are fatigued, they're getting hay fever, and with all that alcohol and the acetaldehyde coming in, that has then affected the methylation pathway, and these gene tests are then becoming relevant. So that would be a typical situation. So the point is that the clinical scenario of the client or patient in front of you is vitally important. If they're FDO positive, that's the obesity gene, but they're looking like they have a waist circumference of women of about 30, or they have a BMI of 22, then we wouldn't start banging on about them being obese. It's inappropriate. It's a risk. So that clinical aspect really is very important, and within DNA health, we do try to be very, very sensitive about that. So, in summary, the genome is the language that your DNA stores to code for who you are. And the epigenetics is the way the music's played. So, alcohol blocks part of your methylation cycle. Exercise will reduce genetic expression of the obesity gene. Smoking in parents increases obesity in offspring. That's all part of epigenetics, and it's an ongoing study, and it's fascinating, and it's very, very powerful. So really my message would be that we can't say that because you are CYP1B2 or 1A2 active or positive that you shouldn't drink coffee. The epigenetic factors around that really are quite powerful, and unless someone is hypertensive, I would encourage them to drink a little bit of coffee and to improve the activity of that enzyme. And within epigenetics, nutrigenetics, so the nutrients that assist our genetics, and this nutrigenetics is where we want to focus the future of DNA and DNA testing, because of the positivity that we can encourage people with that. So that's the end of the first seminar. I hope that was clear. It's always more difficult when you can't interact with the people in front of you, and so it's more difficult for you and a little more difficult for me. So I hope that was useful and positive. And if we have any questions, I know Megan's been typing away and answering questions behind me. Megan, how are we doing with that?
That's a bit of a controversial one.
A controversial one. Is 23andMe reliable for BRCA expression? Yeah, I don't know. I haven't done the 23andMe BRCA expression testing. I have used them and looked at them in other aspects, and I do the BRCA blood test. So I'm afraid I can't answer that 23andMe BRCA test. If caffeine upregulates CYP1B1, 1A2 up. Oh, wait, 2. Let's go back there. All the way back. There we go. Oh, it's got a life of its own now. Okay, so the 1A2. So if caffeine upregulates CYP1A2, as per earlier slides, then would someone with estrogen-linked cancer risk want to avoid caffeine? Okay, let's just, all right, I know the slide I'm going to go to show that. Okay, I understand. So let's go forward here. There we go. So, the caffeine question used to be CYP1A1 until they discovered CYP, and I can't draw this very well. I shouldn't really even try. It's meant to be a CYP1A2. That's awful. That really is terrible writing. A2. So that CYP1A2 is a different pathway. And CYP1B1 is a separate pathway to that. So caffeine should not, from what I've seen, upregulate CYP1B1. But the things that will upregulate CYP1B1 would be the green tea, the flavonoids, resveratrol, and polyphenol. And then the hormone DHEA. Sorry, not upregulate, downregulate, because we want to switch that off. Have people seen that caffeine will upregulate CYP1B1? Because I haven't seen that. So the studies that are generally done are caffeine and CYP1A2. But interestingly, with CYP1A2, other things that go down that pathway are cigarettes and also the charcoal burn toxins that you get from grilling. So cigarettes and caffeine do go down the same pathway, and so you will generally find that people who are cigarette smokers are able to drink their last coffee at 10, 11, 12 at night, and it doesn't affect them because they're so upregulated in that pathway that it metabolizes the caffeine straight away, and they're not kept awake. I hope that answers the question. And if you have got knowledge that caffeine up or down regulates CYP1B1, then let me know. I can go and have a look at that. But I really don't think it does. It's CYP1A2 that it affects. Okay. All right. I think that's it for questions. Megan, thank you so much for typing and answering back to lots of the questions. Bernie, are you still there?
I am, Duncan.
Okay. All right. Okay, so that's great. Bernie, is there anything else we should be going through that I've missed out on? Or should we call that a day?
Just a message to all the attendees this evening. There were a few questions about if they could get a copy of the slides. Tomorrow, the copy of the recording will be sent out. So there should be an email coming out. It'll be from me, and the recording will also be available on the website as well.
Great. And then we need to decide on the date of the next webinar as well. It would also be very interesting to get feedback from people. Was it the right sort of level? Was it too basic? Was it too advanced? Which I doubt. But that sort of feedback would be useful as well, I think. Okay. All right. Okay. Bernie, then I think should we then close up shop for the evening?
Yes, Duncan. Okay. I think that's fine for the evening.